Good day there, students. Prior to this video, I am hoping that you have watched the Punnett square vocab lesson. It's imperative that you watch that to learn about some of the background vocabulary that I'm going to be using throughout this lesson. But today we're going to learn how to use a Punnett square and to calculate the ratio of a specific trait. We're going to talk about what is a monohybrid cross, and we're going to talk about who is Gregor Mendel. So Gregor Mendel was a monk, and he lived in Austria, and he studied pea plants during the 1800s. He studied the specific traits of a pea plant, like height and flower position. He had a background in math and science, and one of his biggest scientific questions was, how are the traits passed from one generation to the next? Through doing this pea plant studies, Gregor Mendel was named the father of genetics. So a pea plant does more than just give you peas. It has many different specific traits. So a pea plant can come in different colors. Some have flowers at the top, some have flowers at the side, so we call that flower position, seed color, seed shape, pod color, and how tall the stem is. So these are all different traits that can be inherited and passed on to different pea plants. And the trait that we're going to use in this lesson is flower color, purple and white. So an experiment that Gregor Mendel did was he took two plants with different traits and he crossed them together, he mated them together and to produce some offspring. So our parent plants was one purple flowering plant, one purple flowering plant and one white flowering plant. And he found out that when he crossed those two parents together, 100% of the offspring were purple. So not a single offspring showed up to be white. So now Gregor Mendel was like, what will happen if I take two pea plants from this generation, from the F1 generation, and I cross them together, I mate them together. So I'll put a little cross in there and we'll mate these two pea plants together. I mean, what would you predict to happen? And it turns out when you cross those two purple parents together, 75% of the offspring was purple and 25% of the offspring was white. So in the F2 generation, the second generation, white showed up again. But in the first round of reproduction, all of the offspring were purple. And Mendel was just absolutely blown away. How is this possible? So what he was able to conclude at the beginning was that the offspring are receiving one factor or one allele from each parent. And this must be the genes that they are receiving from the parents. So a really great tool that we can use to predict the outcome of certain crosses is the Punnett square. The Punnett square is a great tool that we're going to use where we take the genotypes of the parents and we combine them and we can see what types of offspring we will get. Alright? Da 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 da, the Punnett square. It's beautiful, right? Here's a Punnett square playing basketball. This Punnett square just got caught in a thunderstorm. Here's a Punnett square having a joyous campfire in the woods. So the first thing that we need to do before we can use the Punnett square to help us predict the ratios of the phenotypes and genotypes of our offspring is we need to identify the genotypes of the parents. Okay, so what I'm going to use, the letter I'm going to use for my genotypes is going to be an F. Okay, and capital F is going to be dominant because the capital letter is dominant, and lowercase will be my recessive letter. Okay, now first let's look at the parents. This purple plant is from a long line of purple flowering peas. So we know that it is homozygous. It has two identical alleles. And we know now, looking at the first generation, that all of the offspring were purple. So that must mean that purple is dominant. So the genotype for the purple plant is capital F, capital F. The genotype then for the white plant, it came from a long line of white plants, so we know that it's a purebred, homozygous white pea plant. But not a single offspring in the first generation expressed white, so we know that white is recessive. And dominant always beats recessive. So the white genotype is little f, little f. So based on our previous picture, we have identified the genotype of our parents, and now we're going to use our incredible Punnett square to find out what our possible genotypes and phenotypes of our offspring could be. So how we use a Punnett square is we're going to take the genotype from the purple plant parent, the homozygous dominant one. We'll put our capital F's right here, and then we're going to take the genotypes from our white parent, who was little f, homozygous recessive, and we're going to cross them. So first let's look at our, I wrote pink here, it should be purple parent, and we're going to take this big S, and it's going to go in, we're going to put it into this square and this square. Same for this other big F, it's going to, we'll distribute it into this square and that square. Little f's then go here and here. This little f goes here and here. So let me show you. We'll first start with our dominant alleles. This big F goes into there. We have a big F that goes into there. So they came right there. This big F goes into here. This big F goes into here. And they came from here and here. Now we switch back to our lowercase, our recessive alleles. Lowercase. Okay. This little f went here and here. This little f went here and here. Now, from the previous video, we learned that this genotype now, this big F, little f, is called heterozygous because it has two different alleles, a big and a little. And we said that in a heterozygous allele, or genotype, the dominant allele always wins out in terms of the phenotype. So our big F represents purple, and so it's going to crush and stomp the little f so that not a single one of our offspring is white. So this explains why when you take a purple-pink flower and you cross it with a white flower, all of your offspring, all the possible genotypes of your offspring is big F, little f. And in that case, the phenotype then, what we observe is the purpley pink, because that dominant purple-pink beats and wins out and doesn't allow the white phenotype to show up at all. So now what I'm going to have us do, we have identified the genotype of this parent to be big F, big F. We've identified the genotype of the white parent to be lowercase f, and we've identified the genotype of all of these offspring to be capital F, lowercase f, so heterozygous. So now we're going to find out how it's possible that when you cross two of these parents together that have big F, little f, how 75% of your offspring could turn out to be purpley pink, and 25% of your offspring, or 1 in 4, would be white. So in this situation, both of our parents have the genotype for pink. Okay, these are the parents from the F1 generation, and we're crossing them together, and we've already identified their genotype as big F, little f. And their genotype is big F, little f. And all we're going to do with this very simple and outstanding Punnett square is we're going to distribute this f will go into this square and this square. This little f will go into this square and this square. Same for on top. This f will go this square and this square. This little f will go here and here. So I will quickly do that for you. So this square is going to be the square is going to be. So take a moment to observe. Our kind of results of the Punnett square are different. Notice in the top left square we have two capital F's. That is a homozygous dominant genotype, and that's going to result in a pink-purple flower. In our top right, we have a heterozygous genotype, big F, little f, but that's still going to result in a purple-pink flower. Bottom left here, last one, big F, little f, heterozygous. Because the dominant allele will stomp out the recessive allele, we're still going to get a purpley pink flower. But now, whoa, new stuff. Bottom right, we have a homozygous recessive genotype. They have both the alleles for white flowers. So this phenotype would be white. So now we're going to talk in terms of the possibility of offspring. We could say this in many ways. 1 out of 4 would have a white flower, or 3 out of 4 would have a purple flower. We could also say 25% of the offspring will be white and 75% of the offspring will be purple. So those are all ways that we can communicate the phenotype. So these 5 steps here might be a time where you want to hit pause and make sure you have these written down somewhere you want to understand each step. So today we conducted a monohybrid cross. We crossed 2 parents focusing on one trait, mono meaning one. The first thing you need to do to successfully use a Punnett square is to determine the parents that are being crossed, determine their genotypes. Separate the alleles into a Punnett square. Then conduct the cross, determine the phenotype and genotype ratios and percentages. No matter the traits, no matter the parents, those are the 5 steps you'll need to do to successfully use the Punnett square. Taking a look at our last practice problem, we are crossing a heterozygous tall plant with a homozygous short plant. So I like to underline my story problem. I think it's good to write on your story problems to identify what you're looking for. But again, heterozygous, that one's easy. That means they have 2 different alleles. I'm going to stick with using T's because we're talking about tall plants and short plants, but you can really use any letter you want. So we'll make the mom the heterozygous genotype again, big T, little t. And then the other parent is homozygous short. Short here is recessive, and homozygous, meaning they have the same allele. So the father then will be lowercase t, t. I'll quickly conduct the cross. So looking at our genotypes, we can see that these offspring would still be tall because they're heterozygous, but these over here, the phenotype would be short because they only have short alleles. So 50% of our offspring would be tall, and there's a 50% chance that our offspring would be short. You could say 2 out of 4 of the offspring would be tall, 2 out of 4 would be short. And then the genotype ratios, you'd say 2 out of 4 would be heterozygous, 2 out of 4 would be homozygous recessive. If by any means you have questions, we will have follow-up assignments for you to practice on, and use the forums to contact other students or your teachers if you have questions on this material. Once you get a Punnett square down, you can use any genotype and you can use a Punnett square. So once you get it, you're just going to run away with it, and you're going to steal your Punnett square and go start a campfire in the woods. Enjoy, children. Enjoy.